Well, first of all, thank you all very much for coming along today. I'm delighted to be here. I'm delighted to be able to share with you some of our research. And as you can see in the title, I'm going to talk to you about some of the most recent research coming out of my group. And that's on our work trying to understand how temperature regulates flowering responses in breadwheat. And this is a huge topic. And I'm really going to share with you just a slice of the research today. And I put my email address there. And if, as you're listening, you think there's a synergy with the work you do, please do get in touch. We love to collaborate. We'd love to hear from you. So, first of all, why? Oh, I don't seem to be able to change slide.
If you click on the PowerPoint first.
Oh, here we go. Yeah, sorry. So first of all, why are we interested in temperature? And I think the most fundamental reason for that is because actually temperature is a very important signal. It regulates plant growth, and therefore changes in temperature are also leading to changes in how the plant behaves, and ultimately for wheat, that means changes in the final yield. And we're interested in it because it's actually a very kind of cryptic and slightly less direct signal than a number of the other environmental signals. And that's because temperature changes dramatically on a daily basis, but also over seasons. There's a very clear incremental sort of cumulative effect of temperature that the plant is measuring and using to regulate its development. And I think we all know how variable temperature is across a year. And if you were to plot the sort of different temperature patterns between any given year, you'd know from your own personal experience, but also like from empirical data, that the temperature profiles look very, very different. And so we're really interested in understanding how we can use and understand how plants regulate these different temperature or interpret these different temperature profiles to regulate their growth and development. Oh, sorry. Okay, so temperature isn't the only environmental signal that's very important for a plant's growth and development. And the second, or equally important, but the second signal that I'm going to talk about is photoperiod. Now, photoperiod is a much more regular and sort of predictable signal than temperature. And it's obviously used a lot for understanding and dissecting the seasonal responses in plants. In the group, we're particularly interested in understanding how temperature and photoperiod signals link together and how there's synergy between them to sort of reinforce and sort of trigger the seasonal patterns that we see in growth and development. But we're also particularly interested in understanding what happens when the temperature and photoperiod signals diverge. And so you can see in this little schematic down here that under this particular scenario, the winter temperature takes an unseasonably sort of rapid increase. And how does this then go on to regulate and coordinate the plant's subsequent development? And understanding this is obviously becoming more and more important because these particular divergence between the temperature and photoperiod signals are also exactly what we see with the current climate changing scenario. So what do we know at the moment regarding the sort of the genetic and molecular basis of temperature regulation in temperate cereals? And at the sort of fundamental physiological level, we know that temperature regulates pretty much every single developmental stage that the plant goes through. And so right at the start, at seed dormancy, temperature is a very important trigger. And how the plant interprets the sort of temperature signals varies depending on the seed's sort of developmental state. Then subsequently, during germination and the subsequent vegetative to reproductive transition, temperature is involved in regulating both the hormonal and the gene expression signals in these processes. But then subsequently, during growth and the final emergence of the flower, once again, we know that temperature is a very important and defined signal in these processes. And I've really just highlighted on here in red a few examples of the genes that we know to be involved in this, sort of from the flowering perspective. And so I think it's very clear to see that the temperature does have a defined signaling response and is involved in plant development. And as I mentioned in the title, what we look interested in looking at and understanding in the lab is how temperature and therefore also photoperiod are important in regulating the floral transition. And so in cereals, the floral transition really divides into three major stages. The first is that of the very early developmental transition from the vegetative apex, which just produces leaves, to a reproductive apex, which stops producing leaves and starts to lay down the sort of spike framework for the subsequent flower. The next phase is the apex actually going through the developmental sort of stages to make the body plan or the architecture of the cereal flower. And then the final stage is the growth of that particular little flower up through the central stem and its final emergence. And that's what we obviously observe in the field and in the lab. And today I'm really going to focus on this early vegetative to reproductive transition, but I wanted to highlight the fact that there are sort of three major stages, and we're definitely considering all of them in our research. And so in wheat and other temperate cereals, there are really considered to be three major flowering pathways. Vernalization and photoperiod are very reminiscent of the flowering pathways in Arabidopsis, in as much as the concept of the two pathways is the same. So the vernalization is a low temperature triggering pathway, and photoperiod is obviously tracking the amount of day length. The third pathway in the cereals is a very broad and generic class, which is referred to as earliness per se. And this particular group of sort of genetic loci mostly, and a few characterized genes, are basically the responses which are regulating flowering, but can't be put into the sort of the groups of vernalization and photoperiod. And what's interesting and very reminiscent of what happens in Arabidopsis is that these pathways all converge onto a central regulating gene, flowering locus T equivalent in the cereals, which then goes on to sort of trigger that early developmental transition from vegetative to reproductive growth. And so we really started out when I started here at Leeds in trying to dissect the vernalization pathway in cereals. And the reason for that was, I think, probably fairly apparent given my introduction so far, is the fact that vernalization is the only pathway here which is really known and characterized for being a temperature responsive pathway. So what is vernalization? So vernalization is basically the requirement for a prolonged period of low temperatures before that apex is able to go from being a vegetative, only producing leaves, to one that is competent to make the start of the floral development. And in cereals, there are really known to be three major regulators of this pathway. And by major regulators, these are the genes which, when altered, actually enable the growth habit of the plant to change from one that is a vernalization requiring plant or a winter plant to being one that doesn't require vernalization. They're called spring plants. And so the three major loci are Vern1, which is really the central and dominant regulator of this pathway. Vern2, which is sort of generally considered to be the repressor of the pathway. And I should really highlight at this point that Vern1 and Vern2 in the cereals are not the same genes as those identified in Arabidopsis. So don't draw any sort of parallels between those, please. They are completely different genes, in fact. But these two genes are going on to regulate FT, and FT is very similar between all the plant species. And so with the arrival of low temperatures or basically winter, Vern1 expression increases, which basically enables the repression of Vern2 and any other repressors in the network. So we know that oddsock is in that network, but there are other genes as well. And this enables the release of the repression of FT and therefore its expression and the start of reproductive development. So as I say, these three genes are really well known and well characterized. And we're particularly interested in the group in understanding, first of all, how temperature itself is regulating vernalization, and then how we can sort of dissect that temperature response to understand how we could fine-tune vernalization, how we can make it a more predictable response, which obviously would have benefits for in the agricultural setting to enable sort of a predictive vernalization requirement, so people know or can at least anticipate better drilling times and therefore harvest times. And so to sort of dissect this response, it was one of the very first experiments that we set up when we arrived in Leeds. And so I'm saying when we arrived because Kate O'Connor really needs to have a lot of acknowledgement here. So she started her PhD with me last year, and she's responsible first of all for the experimental data I'm showing, also for the very high quality graphics. So thank you, Kate, for those. I should highlight that the lower quality graphics are my own. And so the experiment that we started when we arrived here was one of basically shifting plants between low vernalizing sort of inducing conditions into a glass house that had completely floral activating conditions. And so we ran 3 experiments. The first was 8 degrees under short days, then 10 degrees and 14 degrees, both under short days. And so, as I say, at 2 weekly intervals, Kate was moving plants up into the glasshouse, and that glasshouse was set at 22 degrees long day conditions. And so from this, we are able to identify at which point along the developmental profile the plant was competent to flower and therefore can be considered to be vernalized. And what we observed did surprise us because basically, and I'm just showing the results for one particular cultivar, what we started to see was actually the warmer temperatures were much more efficient than that of 8 degrees. And I think you can see here quite clearly that actually just 4 weeks in sort of the 10 and 14 degree conditions were enough to enable this particular cultivar to be vernalized. And it did accelerate a bit for up to 6 weeks, but by 6 weeks, it was pretty much completely saturated for its vernalization requirement. And so this is much warmer than we first anticipated, and certainly is much warmer than the conditions that we've been vernalizing at before. And so we're pretty excited by this. But what we also observed whilst we were doing this particular experiment was we were dissecting out the apex as we went along. And you can see very clearly here that actually all of these, up until the 10-week point, are still very much in a vegetative state. And so the dogma of the fact that vernalization enables the transition from that vegetative apex to a reproductive one is not absolutely the case. What it enables is a developmental competence to then go on to become a reproductive apex, but that doesn't actually have to translate into being an actual physical and visual change in the apex structure. And so we thought this was pretty interesting. It also meant that obviously the kind of guide that we've been using before to identify floral competence of actually having an apex at double ridge stage, or basically that's the reproductive first stage of the reproductive development, the double ridge, isn't sufficient to actually identify vernalization completion. So we then sort of speculated, well, okay, what we're observing were very different sizes in the little vegetative apex. And obviously for the lines which were then sitting in vernalizing conditions past that 4-week point, they had more time to basically transition through the apex stages and transition and basically become a bigger apex before they're going into floral development. Does that translate into an actual increase in the spikelet number, and so basically an increase in the number of grains or the number of grain potential within the wheat flour? And again, quite surprisingly, what we observed was no, that is not the case. For the apexes which were sitting in a vernalized state at the sort of vernalizing temperatures of 10 and 14 degrees, they didn't go on to then produce more spikelets. And so this is sort of, again, counter to what we were expecting, and kind of raised some more problems or sort of questions regarding what is actually happening developmentally as the plant is going through the vernalization process. And understanding this is particularly important when you consider what's happening in the field. So if we're saying that the plants are actually vernalized after 4 weeks at 10 or 14 degrees, it's quite apparent that plants are fully vernalized by the end of autumn. And therefore, if sitting in a vernalized state does not result in an increase in the spikelet number, and therefore, an anticipated increase in seed production. You can see that actually optimizing drill time becomes more important if we want to make the final yield of the crop more robust. So we were interested, of course, in what was actually happening in that apex and in the leaves that signal into that apex to enable the vernalization transition. And so to start to dissect this, what we've gone on to do is actually measure gene expression across the vernalization time course, and to do this at 3 different temperatures. So first of all, I'm just going to show you the full dissection across the 8-week time course for the 8-degree experiment. I think that you can see that the genes are behaving pretty much as we may expect. So as I mentioned at the start, Vern1 is the activator of the pathway, and it comes on fairly early, and it's a fairly steady increase during the vernalization duration. Vern2, the repressor of the network, is also showing a fairly, relatively steady decrease in expression, and then very slowly you can see a slight increase in FT1. The sharp-eyed among you, you would have seen that the scale on these graphs is completely different. And so when we actually show this in a scaled way, I think actually our understanding changes quite a lot. And what you can see is that Vern1 does show that very steady increase, and it really sort of supports its existence as being this the central dominant major regulator of the pathway. But actually Vern2 and FT1 are hardly expressed at all. And so this really kind of raised the question, well, are these, the major regulators at this stage? And that's an important question, because actually the vernalization genes have been identified through the measuring of flowering phenotype after different vernalization conditions. And so that's the actual emergence of the ear. And I think what may have happened is that some important genes earlier on in the developmental stage have been knocked out by the role of potentially important genes which regulate the later stages of the vernalization pathway. So as I said, we looked at 3 different temperatures, and I think you can see that the genes by and large are behaving in a fairly similar way, not a completely identical way between the 3 temperatures, but I think you can also see that actually the temperatures and the gene expression are not correlative to the flowering phenotypes. And so, we're obviously very interested in trying to understand why that might be and how we can dissect that a little bit more. And therefore, we decided to look at the 4-week time point between 2 temperatures to try and dissect out the response in more detail, and to also try and dissect out the response avoiding the really dominant effect of Vern1. And so that's why we chose 4 weeks, because it's before the Vern1 gene is kind of having a completely, overall developmental response, i.e. it's before vernalization is complete, but it's also at a point where we do see clear temperature specific variation in gene expression and in flowering time, I should say. So in order to dissect this a little bit more, I now need to take you on a bit of a tour of the FT genes, because as I was saying, that you can see quite clearly that the FT was hardly expressed at all, and yet it's meant to be that major central regulator of the flowering time response. And so FT1 had very low expression, arguably no expression. And so what do we know about FT in wheat, and how may we actually use this to guide our subsequent experimental direction? So very similar to Arabidopsis and to rice, FT forms a complex with FD and 1433, which is the floral activating complex. And it's been shown obviously in Arabidopsis and in rice that this complex forms in the little apex, but the FT gene itself is expressed in the leaf, and it travels down to the apex and forms this floral activating complex, which then goes on to regulate floral development. Now, as I'm talking about wheat, I'm sure many of you know wheat is a hexaploid, and so there isn't just one FT1, there are indeed 3, one on each of the genomes, the A, B, and the G genome. And so you immediately get an increased level of complexity there. But actually, it's well known, or relatively well known, that in the cereals there's a much larger number of FG genes out there. And I mean, this is obviously quite a daunting number of FG genes, and there's a large sort of body of research effort and energy now going into sort of characterizing what each of these are doing. And what we know is that some of them are activators and some of them are repressors. A lot of them are completely uncharacterized and we have no idea what they're doing. But also we know that certain genes also have an increase or variation in copy number. And so this means that they're definitely doing something. And so it kind of came to us and to ask the question, well, first of all, why are there so many of these FT genes? Are they actually all FT genes or not actually some other family member? And is this really just a cereal sort of phenomena, or can we actually glean something from other other plant families? And so at this point, I really need to acknowledge Tom Bennett, who at the start of the first lockdown, we decided that this was a great lockdown project to really actually try and get to the base of what is going on with all of these FTs. So I was coming at it from a very monocot perspective, and I was very interested in understanding what was happening within the monocots. And Tom's really brought in a much wider and broader sort of plant perspective. And I think that's really, as so often is the case with collaboration, really strengthened this particular project. And the reason for that is that we can now see very clearly that within the protein family that the FT gene belongs, and so that's the PBPs, that actually there's a very sort of coordinated and reliable increase in the presence of that gene family across, as the plant species increase in complexity. And it's very clear that in the angiosperms, that FT arrives. And so FT can be identified by that little green circle, and you can see here that it is there and present in all of the angiosperms. When you delve down a little bit more into those angiosperm FTs, what you identify is that there are really three, or what we identified at least, was that there really is three major classes. The first is what is classically identified as FT, and so you can see here that's the eudicot FT, and sort of groups with those FT1 and 2 from the cereals. And then there's two other very clear and quite large FT families coming from the cereals, FTY and FTX. And when you kind of go into it in a little bit more depth, what you can actually see is, okay, in the monocots, these larger families obviously exist, but they're not. It's not like there was an explosion. It's very much more there was a coordinated increase in gene number, which ultimately ends up looking a bit like an explosion. And I think you can see that actually by the Poaceae, the gene numbers really have, or the gene family numbers have really got quite large. But interestingly, there is a lot of sort of dynamic regulation between the different plant lineages. And so you can see by the Triticale that some of the families we identified in the Poaceae are actually, they're not present in the Triticale, but they're equally, that they've gone on to compensate by generating more FTs. And so this really does highlight that, first of all, the FT gene is undoubtedly doing more than just floral regulation, but also that there is something intrinsically required in monocots that means that these genes are being held onto. And we decided to try and glean a little bit more information on this by looking at actually sort of the amino acid level of conservation. And so there's been a lot of work in Arabidopsis and rice identifying particular amino acids which are involved in, first of all, the formation of the 14-3-3 base complex, and also identifying the amino acids which are required for either activation or repression of the gene's sort of activity. And what we identified is that, so the FT1 and 2 of the monocots, we could anticipate to behave very similar to the Arabidopsis or the angiosperm FT. The second sort of major clade here, so that's from FT3 to 14, I think you can see that actually that it's retaining most of the amino acids required for a 14-3-3 interaction, and so they're the ones that are highlighted in blue. But actually, when you look over here and look at the sort of the core amino acids involved in whether the gene has an activator or repressor function, it sort of suggests that these ones are more likely to have an activity as a floral repressor. Then conversely, the final group, you can see that actually these are probably less likely to form the 14-3-3 interaction, but are more likely to have an activation role in floral regulation at least. And so this is sort of an interesting insight that, okay, this family has expanded a great deal, but there is clear potential for actual neo-functionalization. So to sort of bring this back to what I was talking about, and you can probably see where this is going now, that we've seen these very clear, expression differences, but we really wanted to understand what were the sort of the minor regulators involved in temperature control of the apex at 4-week time point in vernalization. And so in order to actually do this, rather than trying to delve in and do a bit of a guesswork of qPCR, we decided to go for a more global approach, and we actually conducted an RNA-seq expression analysis for samples comparing the 10 and the 14-degree sample time point at the 4-week stage. And that also showed that FT1 was very lowly expressed. What we did identify was that FT4, so one of those FTs that I've just been talking about, showed a very clear temperature differential in its expression, and that it was actually more highly expressed at the warmer temperatures. So if we go back to that protein sequence that I was talking about and looking at the sort of different potential functionality, you can see that FT4 we could anticipate to definitely form a 14-3-3 interaction because it's got all of the core amino acids to do so. Doesn't mean that it will, but it's got the amino acids to do so. But also quite clearly that it's lacking the amino acids that, or as altered amino acids for the activating domain. And so this would suggest that FT4 is a repressor. And so this kind of brings us to a very sketchy little model that we're sort of trying to elucidate a little bit more in the lab at the moment, is that there is some sort of leaf-based competition between FT1 and FT4, regulating the temperature-based developmental sort of response at that early sort of vernalization time point, and the FT4 is indeed repressing that response at the warmer temperatures. So that really just leaves me to bring the talk to a close and just say that in summary, I hope I've convinced you that vernalization at different temperatures is a thing, and that actually much warmer temperatures are being much more efficient than we first anticipated. The differences in temperature can be distinguished at the gene expression level. And it really does suggest that there are more sort of regulatory pathways than that simple Vern 1, 2, 3 scenario that we have at the moment. I just want to kind of end by highlighting that as a group, we're really interested in sort of dissecting how variable temperature pathways are regulating the different genetic contributions and how this regulates floral development. So this really just leaves me to thank a really excellent team of people that we have here at Leeds. So it's Adam, Kate, Dom, Will and Indy are in my group, and I've just been endlessly impressed with how well they've coped with the COVID situation, and I'm just incredibly grateful that they're here and working on this with me. I'd also like to obviously thank Tom Bennett for his excellent work on the FT project and to really expanding the scope of that particular work. I'd like to acknowledge Steve Penfield and Simon Griffiths at the JIC, who have also supported this very much in the background. And so I just also need to acknowledge the funders and yeah, open for questions. Thank you.
Thanks very much, Laura. That's really great. That FT slide was a bit mind-blowing, actually. And, yeah, golly. Oh. So yeah, let me start with the questions. Let me first of all encourage people to put questions for Laura in the question box there, so I can pass them on to her. So let me ask a question about the FT. So are you starting to look? I mean, this is challenging, but have you starting to use like the new tilling populations that are available to try and dissect what's going on with some mutants of wheat?
Yeah, absolutely. Yeah, I mean, it's a slow game. They're obviously if they're for activators, the tilling mutants are largely late flowering, and so it's an even slower game. But yeah, we're dissecting the response genetically as well as in the lab.
Okay. And let me ask, what was the basis for choosing the 8 and 10 and 14 degrees? Because they seem on the face of it quite close together, but obviously you saw effects there. Why did you go with those particular temperatures?
Yeah. So we wanted to go with temperature. So basically, we want to try and dissect variable temperature responses, and so we wanted to stay fairly close in temperature to actually be able to kind of give some indication as to whether variable temperature we could anticipate to see differences. So that was the first reason we went for close temperatures, and the second was because we want to try and find, and this is a sort of a second project that we're working on, an optimal vernalizing temperature. So that's sort of like developing a speed vern alternative to basically accelerate how much work we can do in the lab, and also for wheat breeders to accelerate how long it takes them to vernalize plants. So it's part of a temperatures series that we're looking into.
Okay, so I just got a message saying the question box might not be working. So if you want to ask a question, please put it in the chat box instead. That is working. I just got a question through from Jill actually. So I've just got one in the question box as well. So, but let me take one from Jill Harrison, who's going to be talking in a minute. Are there any markers for the reproductive transition? Is the spike determinant, and are the number of spikelets invariant or not?
Yeah, so there's a few questions there. So we're developing some markers that we can identify to give us as indicators for the different transition stages, and so we've got a few of those sort of in process. The spikelet number is under genetic control, and so it is fully variable, but for any particular cultivar, then that is under environmental regulation as well as sort of the genetic regulation. So
okay. So a question from Laura Roden. She asks, do you know anything about the spatial expression patterns of the different FTs and how these might compare to the verns?
Yeah, so no. To simply answer the question, no, we don't know. I think it's a really interesting thing. That's definitely something we're looking to sort of work out how we can actually look at all of the different FTs and get us sort of specific enough sort of in situ going to be able to identify the different sort of expression patterns. But no, at the moment we don't have that data.
Okay. Right, the questions are flying in now. That's good. Maybe there was an issue previously. So, so a question from Baran de Graaf. He asks, besides the transition to flowering, do you see differences in grain size or productivity after or between 8 to 10 and 14 days?
Yeah, so that's quite a hard question to answer, and so we do see differences in sort of the spikelet number between the temperature conditions. Whether then that translates robustly into changes in grain size, I can't honestly say, because we've looked at multiple cultivars, but we haven't looked at enough to be able to sort of give a reliable sort of quantification of that. But we don't see, it's not a dramatic change, so we don't see any impact on sort of productivity for subsequent generations.
Okay, so there's a question which I suspect you haven't got to yet, but I'll still ask. So there's a question from Jiffin, and they ask how FTs and verns act to control flowering at different temperatures, whether you see differences in epigenetic changes. Have you looked at that yet?
This is something like we're super interested in dissecting. It's something we've started looking into in the lab, but yeah, we don't have that data at the moment. We are definitely working on seeing how and indeed if the temperature is being regulated at the epigenetic level. Yeah.
So a question from Wendy Moore. This is a bit of a broader question. To what extent may the increase in FT genes be related to human selection since beginnings of agriculture?
Yeah, so that was one of the reasons why I wanted to look at the phenology of the FTs, was actually, do we think that it's down to sort of domestication? So do we see an explosion in FT number in just the domesticated plants? And you can see from the monocots that probably not, but like we can't rule it out, and until we kind of understand what all of those FTs are actually doing, we certainly can't rule it out. So yeah, I think that is a really interesting point. There's definite evidence for in barley, one of the sort of FT family genes has definitely been involved in, it has been selected, and you can see that that's occurred, multiple times. But again, it's really hard to dissect out whether this was selection on like of humans or if it was just adaptation that we've kind of then utilized. It's a tricky question, but yeah, I think that's a really interesting point, and you kind of feel that it must be somehow linked, but yeah, I don't have any evidence in that area.
So this is another one which I suspect you're getting to, but a written question from Richard Mott. He asks, are there any functional differences between the FT homeologs?
Yeah, so I mean, again, so I'm deferring a lot of these questions, but please do take into account that we're quite a new lab. And so basically I don't know, but what we do see is that there are very clear expression differences between them. So between FT1, A, B, and D, FT1B is very much more highly expressed, and that's true for a lot of the different FT genes. And so it does suggest that there is some sort of, at least at the regulation level, there is some differences. And again, you would anticipate that is then seen at a more functional level.
Mm-hmm. A question from Seth Davis, and Seth asks, does temperature act to speed up or slow down the juvenile transition in wheat?
So again, as with any sort of temperature response, this is very developmentally, there's a very clear developmental context to it. And so in a post-vernalized apex, temperature would act to speed up that transition. Whether that then leads to a higher spike plate number, probably not, because accelerated development would basically mean that apex goes through the development stages much faster and therefore sort of reaches the terminal spike plate stage earlier. And that's generally what we're seeing. But for vernalization in itself, so we're seeing very much like a bit of a U shaped sort of efficiency response, and that's been reported historically as well, that there is an optimum temperature that is slightly warmer. So yeah.
Okay, so final question. There are a few others, but I'll pass those that I don't get to onto Laura. There's been a great deal of interest. So final question from James Cochran, and he asks, there's some old reports about red to far red light ratios having an effect on vernalization. Are you doing any work on light quality of that way to look at that?
So I have done that, and we have we don't have any sort of conclusive data in that area at the moment. So
okay, something to be done as well. All right, so I think we'll leave it there. So
thanks. There's a lot on the to-do list, I'm afraid.
Yeah, no, indeed. Well, just when you start a lab 6 months before a global pandemic, it makes it more challenging for sure.
It does, doesn't it?